All right, so we're going to do a continuation. So now we got to the point of understanding the protein synthesis. But what happens if it messes up? What happens that DNA and RNA maybe get in an argument and a letter changes in the midst of that, or during meiosis, it didn't the egg the chromosomes didn't split right because it never connected to the spindle fibers. Well, this is called genetic mutations. What we're going to learn about is that the difference between chromosome and gene mutations, and then we're going to recognize how mutations occur in organisms. Now, any change in genetic material is a mutation, right? So if you are into Marvel, those are mutants, right? That is why if you see them in the end game and everything else, anything that was a mutant was obviously pretty much taken. And the Avengers, look at the Hulk, definitely mutant. Wolverine, mutant. This is called by mutagens. Okay, any agent that can cause a change in DNA. So mutation is a change from the original DNA. Think about it. Was a Teenage Mutant Ninja Turtles mutant? Mutant is because their DNA's changed. Obviously, turtles don't do all that super stuff, right? So this can be random. It can be spontaneous. It can just be random. Like over in your life, it can kind of just change it. And sometimes it can be because of radiation, which is possible, chemicals, or even high temperatures. Now, cells do produce different types of amount of proteins depending on what is needed to maintain your homeostasis. But when changes occur in DNA, these proteins pretty much sometimes are not made properly, right? And so what happens is the overproduction of proteins or underproduction of proteins or proteins just made at the wrong time can lead to cancer or can lead to this. And looking at that frog, I want you to look at that. He's definitely deformed. He got multiple legs, go in different areas. Something's not right. So cancer again is a cell that can't stop, right? So if it's overpopulating and if it's too much, this could just lead to cancer because something went wrong, right? Most of the times, if something goes wrong with the cell, as remember we told you before, it's supposed to kill itself, called apoptosis. So the cell suicide, if it never takes place, cancer happens. And if this kind of stuff in mutants and mutagens take place, if it doesn't get caught, it turns into cancer or it turns into a real issue like this frog. So, if there's a mutation change in the sex chromosomes within an egg or sperm, when fertilization takes place, there will be mutations in the genetic makeup and of the offspring too. Now, we're actually going to go into this um, later about just more different mutations that do occur. That is going to show up inside of your children. But mutations that are autosomal. So remember, autosomal is somatic. And well, remember what somatic is. Those is body cells, right? So autosomal means body, somatic means body. Chromosomes within your body cells will not be passed to your offspring. So mutations in your sex cells will be passed to offspring, but mutations in your body cells will not be passed to offspring. So this is just to give you an idea. So if you have an egg and sperm and a fertilized egg and a mutation occurs, that same mutation is going to go into the reproductive, the bone, pancreas, because it's inside your DNA. A normal cell, once it has it, it's just going to be here, but it's not passed through. So let's talk about the first one. There are different types of mutations. We have a gene mutation, which produces in one gene, so not too big of a deal, but can be a big deal. And then we have chromosomes, which it changes your whole chromosome. Remember, your chromosomes got a lot of genes and alleles, a lot of different DNA. So, just an example of a gene mutation. Uh, we have two types of gene mutations. First one is called point mutation, and this is a change in a single nitrogen base, which is the ATGCs, which may affect the amino acid. So this is normal, right? So a normal blood type, just to say, is GAA connected to CTT, right? Well, if one letter changes, so instead of being CTT, it became CAT. Well, that was actually the code for sickle cell, no longer glutamine. So what happens is, yes, just changing one letter inside your DNA says, hey, sickle cell, come live with me. So another one, basically just examples of the point mutation, is substitution. Like me, I played basketball. It's called, you had a substitution that came in. You would stand on the sideline and you would sub out. If I'm not at school and someone's teaching for me, we call that a sub. It's just a substitution. It's just when something is being replaced. So a single end base is replaced with a different end base. So that's what happened basically for sickle cell, that one letter change. So as you see here, this was the strand, but now look what changed. So this obviously is going to make something happen. Now the thing about point mutations, you have a silent mutation, you have a missense, and a nonsense. Silent means that no amino acid change, redundancy in code. So let's just look here. This is the original strand, okay? Now, instead of here, see this is the C here, it became a U, but that's still coded as glycine. Okay, there's no difference. Okay, that one letter messed up, but it was whoo. That was one of the things that say, hey, we did good. Like, and my name is Shantae. If I take this E and change it to an A, it's still gonna say Shantae. Period. Regardless. Okay, that's good. Now, missense is it changes the amino acid and it changes the final protein. And that's something that you don't want. If it starts changing these proteins, something is obviously wrong. So we go back here and it was Gigi. The A switched to this, and now look, it's a whole different protein. Something ain't right. So that's like me taking my name, Shantae, and then I take my E and I put in a K. Shantique, 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 Shantique. That's not the same. No, not even. Don't even try it. Period. So then we got nonsense, right? So nonsense, obviously, it just changes it to a stop codon. Like if we supposed to keep going and you tell me to stop, that's like if we driving down the highway and it is, I have to get off on exit 50, and you tell me to stop at 47 and make a right, and there's no way that I can get back to exit 50, you stopped me too early for what? So doing that, prematurely, it definitely causes an issue. So the next one is frame shift. This is when is it addition or deletion. So when you're adding or deleting, this obviously changes the reading frame. So here, this was reading cat, TCA, CAG. So if I go here, CAT was here, TCA was here. So something obviously it's obviously been shifted. That means something was added because we was reading cat. We're not reading cat anymore. We're reading over to the right. So that's again like me adding another letter to my name. No. Okay? No. That's just not right. Shantib? Nah, that's not right. So frame shift, it changed because you either move it to the right or to the left of adding or deleting an extra letter. So this is just um, asking you to look at this and tell me what kind of mutation is this, and then what kind of mutation is this. I just want you to look at it and write it down on your notes. So then we have chromosomal mutations. Chromosomes involve, of course, with your chromosomes. This occurs in all living organisms. Most common in plants. And what happens is the meiosis or mitosis in plants, and you'll realize that this is supposed to be a normal strawberry, but look at this. The strawberry seeds. I know some of y'all like them two-butt strawberries, and it has all that juice, and they be real big and red too. Yeah, that's really just kind of eating a mutated strawberry. But what happens if it happens in meiosis and it doesn't separate? So you remember in meiosis, the sister homologous pairs are supposed to separate. Well, this right here never connected. So these two went and had extra. Remember, it's not supposed to be that way. It's supposed to be this is they pulled apart. This ended up going with the sister. It was like, oh, I didn't want to leave it. It was just like tied up. Now let's bring extra cells. Extra cells is obviously going to be what we call Down syndrome. And again, we're going to learn about that. Um, Down syndrome. And Down syndrome, and this process is called as non-disjunction. And the way that I say it is non-disjunction cannot function. People who have an extra chromosome cannot function as well as people who have um, the normal chromosomes. When homologous chromosomes fail to separate in meiosis, so again, non-disjunction, the biggest example is Down syndrome because they get an extra chromosome. So here's the types of chromosome. We have deletion. So part of your chromosome is left out. Like it must be A, B, C, D, E, F, and you talking about AC. Like where to be at? So then we have insertion. So this is just common sense. If you're deleting something, obviously you delete. If you're inserting something, you're adding something. So this is where it gets. A part of the chromatid breaks off and goes to another chromatid. So one of them is getting deleted. So that B that was over here said I wanted to come down here. And then we got inversion. So I play music, and inversion is just basically taking your notes to another octave, or just rearranging the note level. Um, but when you do this, it's basically supposed to be A B C D E F, and it became A E D C B F. But you're like, hey, uh, same letters, yeah, but you did it backwards. Something's not right. Then you have translocation. Trans means change. Okay, so anytime you see trans, it means change. And then you have location. Obviously, this means we're going to change location. So the chromosome breaks off and it's added to a different chromosome. So can you imagine a piece of your chromosome? This is right here, the inversion, where it says a part of the chromosome breaks off and is reinserted backwards. So can you imagine something being broke off and then coming in here and translocation can change the whole thing? Like, look, it's supposed to be A, B, C, D, E, F. It's A, B, C, J, K, L. Somebody, somebody get that. That's not right. So if you look here, you can actually see, hey, we're missing, so that's deletion. We have an extra, that's obviously um, insertion. Now look at here, we done switched, so this can be inversion. Now I wanted you to see this because this is just a better way for you to see it with using a name. So look at here, this is normal, it's supposed to be beast, but if you change that one letter, subbing it out, it changed to a feast. Well, let's say we added another letter. That became a chicken breast. Let's say we take it off. Now it just says best. And then we have inversion. Now it has beats. So we have all of this to look at as an example. There are some advantages. One of the biggest advantages was that DNA was changed. Um, the amino acid sequence was changed. The new protein form changed your phenotype. We haven't learned about phenotype, but it is our next unit, which means your physical appearance, how you look, which increases your chances of survival. So the DNA was changed, the amino acids sequence was changed, and as you see here, there's a lizard in the ground, little, I don't know what kind, I'm not really into into those. And there's an owl that is inside the tree. So with that being said, that's an advantage. Housing from the prey, physical appearance. We also can DNA repairs itself. So like me or your phone or Microsoft Word, it catches do a typo if it finds the error in the nitrogenous base. So most of the times it's supposed to fix the error, but sometimes it's not perfect. Your phone's not perfect because sometimes it may autocorrect to a different word, and that's greater exposure to a mutagen, and it's more like the typo will not be corrected.